It's a great pleasure to be here. I've had a fantastic day talking to folks about research, and yesterday too. My brain is really cooked from all of the fantastic stuff I've heard about. Um, but today I want to introduce you to a totally different kind of model organism. It's not a fly, and it's not a nematode. It's a polychaete, which is a kind of an annelid, a segmented worm that we're developing as an experimental model for developmental evolution. And so I've picked this sort of aggressive title that developmental evolution is a population genetics problem in order to motivate why this new weird model organism is something worth studying. And so when I talk about the evolution of development, I'm not really talking about like the origin of phyla and these kind of deeper aspects of organismal diversity. I'm really talking about like the process of development from an embryo to an adult. Are there things about that process that are special that we need to consider when we're doing population genetic modeling or analyses? And the claim I want to make is that indeed there are several things that are really special about this process of development, this ontogeny from a single cell to a complex adult, that we should take into account. And I like to motivate this with this classic figure from a paper from many, many years ago now, from Albert et al., 1979, in which the authors ask us to imagine development is basically a trajectory. And so here we've got this sort of conceptual space that involves an age axis and a shape. I guess this is a size axis here, and this is a shape axis here. Shape is, of course, some hugely multivariate, complicated thing, but we can imagine compressing it down to one axis for visualization here. And in this case, a single individual's phenotype is this trajectory through the age, shape, size space that gives us at the end some kind of adult phenotype. But to get to this point from the starting point, there is some shape that it follows through time. And each individual has this trajectory, which means that at a population level, what selection is really acting on is variation in these trajectories. And so at any particular moment in time, there's a distribution of phenotypes, but those are then genetically correlated with the distribution of phenotypes at some earlier time. And selection acting on any one of these times is really selecting among these trajectories, which is going to have sort of pleiotropic genetic correlation effects on the other points in the trajectory. And so this is one aspect of development that we want to consider. And if we think about now selection acting on a particular adult phenotype, like, you know, canonically, here's some Darwin's finches. Selection is acting on their beak shapes right here. So there's some natural selection happening, ecology interacting with these phenotypes. That selection is then also moving this trajectory down here as just a byproduct of selection on the adult. So there's this pleiotropic effect upstream in development. Of course, most early development is happening inside an embryo, which is itself the product of an oocyte. So this big thing that's full of maternal cytoplasm. And often this will be protected from the environment during this early phase of development. So for example, we can imagine a bird egg where there's lots of development happening inside a shell. And so in this case of the birds, selection is acting on this phenotype here in the adults. And early development is happening sort of isolated from that ecological environment. And so conditional on the embryo emerging from the egg at this point in the age, shape, size space, anything that happens inside this egg is kind of shielded from ecology in some sense. It's happening inside this separate environment. And so, so evolution that changes stuff in here is kind of irrelevant, conditional on it emerging right here. And so we might imagine that evolution could happen by changing the amount of time or size or shape that's happening inside this sort of isolated setting. We could also imagine evolution going the other direction, making a kind of smaller protected thing, which then allows for a partitioning of resource use into a larval stage, where now selection is going to be acting on some kind of phenotype early in development that's totally unrelated to the phenotype later in development. And now we have this potential for pleiotropy restraining the independent evolution of these different ontogenetic phases. And so the first thing I want to say about development is that it forces us to think about these onto genetic correlations, where things that are happening at one point in development are shaping things that are happening at a different stage of development. And of course, there's environmental effects that happen early that affect things late, but there's also evolutionary effects where things happening late in development are influencing things that are happening early in development. And so that's one aspect that I want to emphasize. But the other is that the egg, where all of this sort of cytoplasmic stuff is happening here, is also a phenotype, but it's a phenotype that's produced by the adult. And so for all of the stuff that's happening early in development, we have to be thinking about the genetics not just of that embryo, but also of that embryo's mother, which is responsible for making that starting material. So maternal genetic effects are really, for me, what's exciting and special about the population genetics of developmental evolution. And there are a bunch of things that are really special about maternal effect genes. These are the genes that are acting in the mother to affect the offspring. One thing is that these are sex limited in their expression. They only affect phenotypes when they are present in females that are producing eggs, but the genes themselves are also present in males. But the selection only sees the female phenotype. And that means that a kind of mutation selection equilibrium, these genes will harbor twice as much genetic variance as a gene that's expressed in both parents. Another feature is that selection is always acting indirectly. It's acting on the indirect genetic effects. And so the phenotype that selection is seeing is the offspring, but the individual who's carrying the maternal individual is expressing the phenotype, but the offspring are carrying the genotypes that are getting transmitted. And maybe I'm saying that exactly backwards. But you see that there's an indirect relationship between the phenotype that is under selection and the individual carrying the genotype that expresses it. And the most important thing for our purposes has to do with the way that maternal genes create the environment in which zygotic genes are expressed. And so here we're considering the very simplest case of a single locus that acts both maternally and zygotically. And we can imagine now the offspring have these possible genotypes at this locus, but each of these genotypes, for example, this heterozygote, it can find itself in any of these different maternal environments. So a particular zygotic genotype will be present identically in these zygotes, but these zygotes themselves have different environments as a result of the maternal effect. This creates a frequency-dependent genotype environment interaction, because the frequency that these identical genotypes are experiencing in their environment is a function of the allele frequencies at this locus in the population. If little a is really common, then a lot of the heterozygous AA zygotes will find themselves in this blue environment. If the little a is rare, they'll find themselves in this environment. And that creates this dependency on the allele frequencies, but the environment that the zygotes experience. And this has lots of downstream consequences. So if we want to think about how these factors are influencing phenotypic evolution and genetic evolution, then we need model organisms that vary in the appropriate phenotypes that will allow us to interrogate these interactions and potentially their frequency dependence. The best organisms for this kind of question, I believe, are benthic marine invertebrates. And so here are some sea urchins. And if you think about a sea urchin, you can imagine its kind of life cycle, which is that the adults live on the seafloor. They don't move a whole lot. And then they release their gametes into the water column where there's fertilization. And then there's a larval phase in which the larvae have a completely different body plan from the adults. They swim around in the ocean and they eat and they grow and they put on biomass until they undergo a metamorphosis and then transition into this benthic adult life stage. This kind of bentopelagic life history is super common in all sorts of different lineages of marine invertebrate. And so here we're looking at, here's a sea star and this is a sea cucumber. This is a urchin. This is a sea cucumber. This is a sea star. This is a ribbon worm, and this is a polychaete. And these all have these planktonic larvae that are swimming around and have body plans that are very different from the adults. And they go through metamorphoses to achieve their adult benthic lifestyles. In each of these lineages independently, and many, many times across the phylogeny, there have been transitions to direct development in these invertebrates. And so here's a close relative here of this animal is this animal. The scale bar is different now. These are much larger embryos, and they're full of yolk. And these animals can directly go through metamorphosis without ever going through this planktonic larval stage. And so we have names for these kinds of larvae. The ones that live in the water column and eat plankton are called planktotrophs. And the ones that develop directly and can skip that planktonic phase are known as lesithetrophs. That means that they eat yolk. So they're dependent on this maternal provisioning. And their mothers, you can imagine, are making very, very different egg sizes and provisioning those eggs very differently. And that then leads to these much larger offspring that have these totally different anatomies in different life histories. So we'd love to study how evolution allows for transitions between indirect development and direct development. And again, this is a transition that has happened huge numbers of times across the phylogeny. So it's very consequential for thinking about the evolution, very broad scales of animal diversity. The model organism for studying this is a polychaete annelid called Streblospio benedicti, which you see at the top. It's this beautiful little worm. It's segmented. It's got bristles on each of these segments, and it's got 4 little eyes. It's got these rabbit ear kinds of appendages here called branchii that are effectively gills. And it's got these 2 long tentacles from its head that are palps. Those are covered with rows of cilia. And this animal basically sits in the mud and puts its tentacles out onto the mud or up into the water, and then just conveyor belts along these rows of cilia. It moves particles of mud, benthic mud, into its mouth, and then it eats the mud. So that's how they feed. Many years ago, here in 1984, Lisa Levin discovered or reported that this animal, Streblospio benedicti, occurs in different developmental morphs, where some of them produce direct developing large eggs, and some of them produce indirect developing small eggs. But it's all one species. And that means that we can do genetics to try to figure out how this transition is possible and what maintains the alternative morphs within a single species. This species has separate sexes, males and females. Unlike the sea urchins, they have internal fertilization, and specifically the males deposit spermatophores on the mud. Females pick up their spermatophores and then use the sperm for fertilization later. And then the females brood embryos and release them after embryogenesis when there is a kind of a free-living animal ready to emerge. And so in some cases, those free-living animals are large juvenile worms that can go immediately into the benthos. In other cases, they're tiny planktonic larvae that have to swim around and eat plankton until they undergo a metamorphosis. Here is a little video to highlight how spectacular these animals are. This is a male waving its palps around, and it's actually holding a spermatophore here in between its branchii, and it's going to release that spermatophore, sliding it along the cilia on its branchii. There it goes. And then a female will come along and pick that up and use those sperm, some cases weeks later. And that's basically what they look like. And the nice thing about this is that they're very easy to grow in the lab. The females then, they fertilize the oocytes, they transfer the embryos into little brood pouches. And so here's a kind of a side view through a piece of a female in which the head would be over to the left. This is the intestine here in orange. And these are some of these big yolky oocytes inside the body cavity. And then in these pouches along the animal's back are these developing juveniles that the female will release into the water column when they are fully developed. The alternative larvae look like this. And so in the lesithetrophs, there's a relatively small number of eggs that are large, and the juveniles emerge already with the kind of head appendages, the palps and branchii already budding out. And so these guys are just ready to go right into the mud. In the planktotrophs, a female will produce many, many more of them, and they're individually much smaller, and they produce this much more complicated little larva that has these rows of cilia around its head that it uses for swimming. It's got these defensive bristles. It's got these little tail appendages. It's a much more complicated little animal because it has to do so much more in nature. And so you can see that there's a trade-off between size and number in terms of maternal production of offspring here. These bristles are only present in the planktotrophs, and they're thought to be defensive structures, basically. And there's a little bit of evidence that they are, in fact, defensive structures. Here's a beautiful video I'm going to show you of a different species of polychaete. This is Platanarius, in which the larva is going to defend itself from attack by a copepod by flaring its bristles, and it gets away. And in this study, the researchers genetically modified the larvae so that they could not flare their bristles. And here's what happens. Unsuccessful in its defense. And so that's why we think these bristles are defensive structures. And the bristles are one of the many traits that had to evolve a difference in this larval anatomy between these two types. So we want to understand genetically what's going on to make these two life histories different in these two species. So we can study these animals right in New York Harbor. Here is New York. This is Manhattan over here. Here's the World Trade Center. Here is the Statue of Liberty. NYU is right here, actually. And we can collect these worms here in Newark Bay and right over here in New York Harbor. And at most places, we only find one type or the other. So at Newark Bay, we only find the planktotrophs. But here at Bird Point, we find a mixture of planktotrophs and lesthetrophs. And we can go out and they're just in the mud. We can go at low tide and collect just by walking into the water. They're also subtidal. They're just everywhere in the mud on the east coast of the United States. And they're also incredibly abundant. They can be in the many thousands per square meter. They're probably the most common animal in mud along the east coast of the United States. So here's a PhD student, Jackie Jackson, sieving the mud to find these animals. And we can bring them into the lab and raise them in 6-well cell culture dishes. They're great lab organisms because they live in these mud flats that are sometimes exposed at low tide. And that means that they don't really care that much about salinity because they experience the whole range of salinities from basically rainwater to hypersaline in the hot sun at low tide. And they don't care that much about temperature because they're out there all year long, sometimes under the ice, but also baking in the summer. And they also don't care about pollutants because they live in the most disgusting places on the east coast of the United States. Newark Bay is notoriously polluted with everything you can imagine, and they're just fine. So here's a typical culture. We give them sterilized mud, and they're just pulling the organics out of that by passing it through their gut. And so they make little mud tubes, and then they eat the mud, and then they defecate out these little rods. And in the course of about 5 days, they'll eat all of this mud, and then we change the mud, and they're very happy in the lab. So we start here today with some things that we already know about these animals. We know that the larval form is highly heritable and not plastic. And so we know this from lots of common garden experiments, and we know it from genetic studies that I'll show you, that basically the variation that we're interested in here is heritable stuff that we can dissect genetically. Second fact is that most localities are homogeneous. They've only got the big type or the small type with some minor exceptions. And finally, we know that these populations are exchanging genes and that they're really part of a single species. And we know this from lots of population genetic data. And I'll show you genetic data a little bit in a moment. Before I get into the genetic results, I want to motivate a kind of ecological model of why these two things exist, why in some places it's better to be a lesthotroph and in other places it's better to be a planktotroph. And this will help us think about how the evolutionary consequences of maternal zygotic genetic interactions are consequential. And so we can start our simple model by assuming that mothers in this system have a quantity of resources that they can invest in offspring. And if we measure the size of each larva in terms of units where 1 is the amount of material necessary to make a lesthotroph that can skip the planktonic phase, then a planktotroph has a size that is smaller than that in some way. So in this case, if the size of the larva is 1/8 of a lesthotroph, then the maternal investment is going to result in 1 larva per unit maternal investment in a lesthotroph and 8 larvae in the case of a planktotroph. But they're not all going to survive, and that's because the water column is a very dangerous place. And so while these animals are pre-metamorphic, while they're out there in the water column, they're going to be eaten, and so the numbers will decay as a function of the predation rate per unit time times the time it takes for them to reach metamorphosis. The time it takes to reach metamorphosis depends on how big they are. If they're almost full-size lestrotrophs, then it's going to be a very short time to metamorphosis. If they're tiny, it's going to be a long time. They're going to have to feed on the plankton for a long time to get big enough to go through metamorphosis. So we're going to say that their size scales the time to metamorphosis. So here's what we might expect. But there's lots of other variables in here, lots of other parameters in this model as well. And so, for example, these larvae are defended by their bristles. These guys are not defended. And so we might suppose that the predation rate is itself scaled by the level of defenses. So we're going to multiply the predation rate by 1 minus something that says how well protected they are, which is how much they've invested in defenses and how effective defenses are. Defenses aren't perfect. A whale doesn't care if you've got bristles, it's just going to eat them anyway. But they can protect themselves to some degree. On the other hand, defenses don't come for free. If you're investing in defenses, you're making fewer offspring. And so we're going to scale the number of offspring a little bit by the cost of defenses. And that cost of defenses is some fixed thing that we can then multiply by the investment that is being made in defenses. And so we're left with this kind of model of how many offspring we expect in the lesotrophic case and how many in the planktotrophic case. And I think an important feature here is that some of these terms are about the environment. How much plankton is there for them to eat? That governs how long it takes them to reach metamorphosis. How many predators are there out there? That governs how much they're being eaten per unit time. But other features are aspects of development. And so the size of the embryos and the amount of investment in defenses, those are things that these animals can evolve. And so if we simplify this here, then we're left with basically two evolvable parameters: the investment in defenses and the size of the offspring. And so this gives us basically a two-dimensional phenotype space onto which we can now plot fitnesses using this, this model that we've just made. And so here's just a very simplistic representation of what this model gives us, where if we pick particular values for these parameters, then we end up with a fitness landscape. In this case, an undefended lesotroph has the highest fitness. And if we vary these parameters, you'll see that the fitness landscape also varies. But certain things are in common across the whole of the parameter space. And the main thing is that there are only two peaks that ever occur, and those are in the corners. So a defended planktotroph can be good, and an undefended lesotroph can be good, but the other corners in the intermediate space are never optimal. And so the fitness landscape here says basically you got to be at those two corners, and which corner is better will vary as a function of the environment. So if there's a lot of predation, then it's better to be a lesotroph. If there's a lot of plankton to eat, it's better to be a planktotroph, because then you have a female will have lots more successful offspring if she makes more of these small embryos. So somehow evolution has to get from one corner to the other, and we can imagine lots of different genetic architectures that could facilitate this. We could imagine a single locus that's pleiotropic that just moves directly from the planktotrophic ancestor to the lesotrophic descendant. But we can imagine kind of any other genetic architecture as well, any number of loci, any degree of pleiotropy between them with respect to these two traits of interest. We can also think about the maternal and zygotic component. And so we might suppose that, I mean, we really know, in fact, that the egg size is a maternal trait. But we can imagine that this aspect, the defenses, could be maternal as well. The maternal cytoplasm dictates how the embryo develops, or they could be zygotic. And those things have different implications for the frequency dependence of the zygotic maternal genetic interaction. So we set out to figure out what is the true genetic architecture in this system. And so we start by doing crosses, and our mapping crosses started with a lesotrophic male from Long Beach, California, This is an introduced population. These animals are native to the eastern United States, and this is an introduction to the western coast of the United States. Cross that with a planktotroph from New Jersey, a female. And so these animals are going to produce small larvae because the egg size is a maternal trait. And so we get F1s. These develop on small eggs. When they grow up, they make intermediate-sized eggs. So that's telling us that there is a maternal regulation of the egg size, and further that there's some level of additivity so that we get this intermediate that we don't observe in nature when we cross them. They make F2s. The F2s all develop on these intermediate-sized eggs made by their F1 mothers. When they grow up, they make diverse-sized eggs, and that's because the maternal genetic stuff that influences egg size has segregated now, and so the F2s make, some of them make big eggs and some of them make small eggs, and they in fact make this whole range of intermediate-sized eggs, which again are never observed in our studies in nature. And finally, when we get to this third generation, now everything is segregating. We've got these animals have grown up on different sized eggs, and they have their own unique genomes as well. And so now we can ask, how do the different zygotic, how do the different larval anatomy traits segregate in this context? So I'm going to focus on these larval chaetae, which I've shown you are defensive structures, and they vary in nature between a whole bunch of them that are very long in the planktotrophs and 0 of them in the lesothotrophs. And also we've got these bacillary cells over here, which are these great big cells that have these rod-shaped inclusions like a bacillus. And these make these tail appendages, and they're called anal-cirri, these appendages. And these are only found in the planktotrophs, never in the lesothotrophs. So we did a series of crosses that resulted in 264 F2s that then produced 4,000 third-generation larvae. And then we can jointly analyze the larval and adult morphology of the F2s and the larval morphology of the F3s, and then put these all together into one big statistical analysis that separates out maternal and zygotic genetic effects. And in fact, we look for maternal and paternal and zygotic. And this is published already a little while ago. So what we found is that these bristles and these tail appendages are inherited in a purely zygotic way, no evidence at all for any kind of maternal genetic effect, any kind of paternal genetic effect either. And there are very large effect QTL that explain more than half of the variation in each of these traits. So the number of these bristles has QTL on chromosome 9 and chromosome 3. Chromosome 3 also affects the number, and so it's chromosome 8. And these appendages on the tail, the anal-cirri, are explained in large part by a chromosome 5 QTL. And again, a lot of these QTL are quite large. And if we look at all the different genotype classes, we can find dominance and we can find epistasis. But basically, a small number of loci are explaining a lot of the variation in these traits. When we look at egg size, we find different QTL, mostly different chromosomes. And again, this is purely parental. There's no zygotic component of this at all, which is, of course, what you would very much expect. And some of the effects are quite compelling. They're not huge. These QTL explain a smaller fraction of the total difference between planktotrophs and lesothotrophs. So we have purely maternal effect QTL on chromosomes 6 and 7. And then we have these weird interacting effect QTL, where there's a maternal effect and a paternal effect that is perfectly antagonistic to it, and no zygotic effect at all. And so this is kind of surprising. We don't really expect paternal effects here, and we don't know the mechanism for this. It's probably got something to do with the spermatophore containing chemical cues that influence oogenesis in the females. That's our kind of best model for what could be happening here. But we don't know the mechanistic details for this. Okay, so that's our quantitative genetic analysis. And now we can go ask, what do these alleles look like in natural populations? And can we use natural population genomic data to refine the intervals around these QTLs? Our genetic map is quite crude, got not that much recombination, and so our QTLs encompass many megabases of DNA. We'd love to let nature do the recombination for us, narrowed down these intervals to small regions where we can say, here's the gene that's responsible for the bristles, for the tails, for the egg size, and so forth, and ask how those then contribute to the variation in nature. So we've got data from Newark Bay. We've got data from Bird Point. We've got data from New England. This is southern New England. This is Providence, Rhode Island up here. So we got planktotrophs from near Providence. Here in southern Massachusetts, we've got planktotrophs and lesothotrophs. This is Woods Hole, which is the most relevant biologist reference point here. And so some of these places are really lovely. This is in southern Massachusetts, where a PhD student, Connor Gilligan here, is collecting some animals in the mud. And I've just actually discovered, I'm sorry to say that somehow I've misplaced the slide that gives credit to the people who did all this work. I really thought that was in there. Connor Gilligan is responsible for all of the population genomics I'm going to talk about. And Christina Zakis, who was a postdoc and now has her own lab at North Carolina State, is responsible for all of the quantitative genetics that I just showed you. And I apologize for not saying her name earlier. So Connor went and collected lots of animals and did lots of phenotyping. And so here, for example, are data from, this is Bird Point and Newark Bay. Those are our two spots near New York City. This 100-acre Cove and Horseneck Reserve, which are our spots in Massachusetts and Rhode Island. And you can see that just phenotypically, there's very discrete categories of larvae here, small and large in terms of their area. And Connor tested whether these animals could directly go into the benthos, and so are lesothotrophs, or whether they required food and could not directly metamorphose. And the colors here actually represent that separate phenotype. So the colors and the sizes are separate things that Connor measured. And so indeed, we see this discrete classification into planktotrophs and lesothotrophs and not intermediates. And so Connor then went and generated whole genome data from lots of other populations. And so I've told you about Bird Point and Newark Bay. He also got Pelham Bay Park in his home borough of the Bronx in New York. Here's our New England locations. Long Beach, California, I have plotted in Chesapeake Bay because we think that's probably where those animals that ended up in California as introductions originated. But we don't know that for sure. And then we've got data from way down here in South Carolina at the Baruch Marine Station, which is very far away. And at Baruch, we also have a mixture of planktotrophs and lesothotrophs co-occurring at the same place. And so a few years ago, Christina Zakis assembled and annotated a reference genome. It's about 700 million base pairs. There's 10 pairs of autosomes and 1 pair of sex chromosomes, as you've already seen in the genetic map. And so Connor generated data from 83 individuals from across this whole range, very deep sequencing of individuals. So everybody's more than 30x coverage, and he can call many millions of SNPs. And when we just look at the principal components of these genotypes, we see that it's pretty stark. That independent of geography, all of our planktotrophs are over here, and all of our lesothotrophs are over here. And so we basically have principal component 1 is separating everybody by morphology, and principal component 2 is separating everybody by geography, and mostly really separating the Baruch population in South Carolina from everybody else. And this is consistent with what we know about marine invertebrate population genetics in general, that there's a pretty big biogeographic barrier here in North Carolina that separates northern and southern. And so there's presumably been less gene flow across this boundary for quite a long time. And so the main thing here is that this morphology axis tells us that independent of geography, there is a bunch of genetic differentiation as a function of the developmental mode. And we've already seen from the QTL mapping that there's a handful of large effect QTL that explain some of the anatomy and the egg size. And so we might expect now to identify from differentiation those QTL loci. And so Connor looked at FST, ignoring geography now, just pooling all of the leucotrophs, all of the planktotrophes. We're just interested in those variants that are perfectly correlated with these morphologies. And across the genome, FST is very, very low. The mean is 2.5%, and the data set that we're looking at here is 17 million biallelic SNP sites that have perfect data that is confident in genotypes in each of the 83 individuals. And I'll tell you that the genome as a whole has something like 100 million variants. And so we're filtering the data incredibly stringently here just to look at things that are perfectly genotyped. That means that we're going to miss a lot of stuff that, if there's segregating indels, we're not going to be seeing those things. If there's places that have read mapping challenges, we're not going to see those. So this is a kind of a first glance at what's the very best quality SNP data telling us about differentiation between these populations. And I'm building up to this plot, which says that differentiation between these morphs on average is 2% down here. But there are these enormous peaks of high differentiation on every chromosome. And that wasn't true for the QTL. And moreover, there's multiple peaks on every chromosome. And so our QTL pointed to a small number of things that are important for this suite of traits that differentiates planktotrophs and lescotrophs. But the genomics are telling us that we are missing a ton of genetics. And part of that, no doubt, is that we're missing a ton of phenotypes. And so when we look at the genes underlying these peaks, we get a bunch of clues as to what kinds of things we're missing. And a lot of them, at least tentatively, are yolk things that have to do with the quality of the material that the mothers are providing to their offspring. And so in retrospect, that's very obvious that the yolk has to be very different in a lescotroph and a planktotroph. And so some of this differentiation that we're seeing is due to that yolk stuff. But some of it is no doubt due to the traits that we've already mapped. If we zoom in on some of these peaks, we'll see that they are in general pretty narrow. And so here's one on chromosome 5 that represents just about 20 kb of stuff that rises up to FST equals 1 across this is more than 40 individuals of each of these two morphologies, perfectly differentiated by these SNPs. And over the whole genome, there's, I think, something like 900 of these FST equals 1 SNPs. And here I've got genes annotated at the top here. You see this is just a couple of genes. So we're in principle in very good position to annotate these variants and start connecting molecular traits to the phenotypes that are involved in this transition from planktotrophy to lescotrophy. In this case, if we look at this small number of SNPs that are perfectly differentiated, they're all here in exon 7 and in the introns and 3' UTR of this gene. And when we zoom in, there's in that interval, one of the perfectly differentiated SNPs is actually a very dramatic mutation that renders a RAB family GTPase inactive, according to most annotations, according to all annotations of RAB family GTPases. So this is one where we've got a potential candidate variant that is effectively breaking a gene's molecular function, and now we can start digging in to try to understand what's happening there. But the annotation of all of the genes, the molecular variants and so forth, that's a very challenging business that requires lots of annotation of genes, and also the translation of gene annotations from other organisms onto our polychaete. Annelids in general are very poorly studied from a molecular biology perspective, and they don't have any close relatives. So when we're looking for annotations, we generally have to go to species that differentiated in the Precambrian, right? So Drosophila, C. elegans, the common ancestor of those guys and annelids is hundreds and hundreds of millions of years ago. So our gene annotations are not great. And so that's not going to be our emphasis. Our focus is going to be on trying to figure out what happened from the population genetics perspective to allow for this kind of transition. And so I'll illustrate that with another peak. This is one on chromosome 3, where the interval is, again, just a few tens of kb, and there's a couple different genes in there, and we don't know which one is involved. But the question we can ask now is, what are the population genetic signatures that coincide with this region that could help us understand what's happening here? And so one thing we can do is just ask whether the lesthetrophs and the planktotrophs have similar patterns of genetic diversity across this interval. And when we look at heterozygosity within each of these populations, we see that there's actually a quite striking difference. That planktotrophs basically don't show any change in genetic variation across this interval. But the lesthetrophs, that's the big egg direct developers, they have a dramatic deficit of heterozygosity here. Polymorphism basically goes away. And so this superficially looks like there has been a selective sweep in the lesthetrophs where an allele that contributes to a lessitrophic phenotype is swept through, eliminated the genetic diversity there, and we're left with an area where there's SNPs at FST equals 1 and a unilateral, basically single population, single morphology loss of genetic variation. And that's at this one locus. We look at the rest of the genome, we find that planktotrophs and lesthetrophs don't really differ at all from one another in their kind of global distribution of pi. But if we plot it as a function, oh, I'm sorry, I forgot the axes here. This is nucleotide diversity within lesthetrophs or within planktotrophs. And this is FST in 5 kb bins. What we find on this chromosome and on every chromosome is that at low differentiation, heterozygosity is the same in the two populations. But as differentiation increases, lesthotrophic pi plummets. And so they basically, every locus looks like the locus that I just showed you, where the planktotrophs haven't really changed at all, but the lesthetrophs have experienced what looks like a selective sweep. And so this is consistent with what we sort of imagine is happening across the diversity of marine invertebrate lineages, that ancestrally there were complicated larvae that were swimming around in the water column, and that in each of these separate transitions, there's been a shift from planktotrophy to lesthotrophy. So this pattern of reduction of heterozygosity, specifically in the lesthetrophs, helps us kind of confirm belief about the polarization of this transition, that we start with planktotrophs, then selection favors lesthotrophy in these other populations. So here's a juvenile that has undergone metamorphosis and is about to go into the water, into the mud, and gives us another glimpse of how beautiful these animals are. And so the basic picture we're left with is that at some point in the history of these animals, planktotrophic species spun off some lesthetrophs. Those lesthetrophs now are maintained in many different locations, and gene flow homogenizes most of the genome, according to our general impression. But these couple hundred different intervals across the genome are maintained by very strong selection to be differentiated between the lesthetrophs and planktotrophs. So it's a basically polygenic pattern where what at first looked like in our QTL data, a few large effect loci, when we treat the syndrome as a whole, we find that there's hundreds of loci and that selection is acting on all of these incredibly strongly. I'm realizing that I have cut out the slides that would then tie this back to the population genetics theory that I started with. I imagined that I would run out of time. So I'm just going to tell you that when we incorporate this genetic architecture, or any of the theoretical architectures I started with, one supergene or two independent loci or two pleiotropic loci, any of these genetic architectures, when we ask what would it involve, what would it require for a population to switch from planktotrophy to lesthotrophy, or from lesthotrophy to planktotrophy, given our fitness landscape, the pattern that emerges is positive frequency dependence that is governed by this maternal zygotic interaction phenomenon. And so the basic story is that if a mudflat is full of planktotrophs, everybody's a small-egged individual, and a lesthotroph moves in, its alleles are going to find themselves in a small egg. And so the zygotes that emerge in that next generation are all going to have small-egged mothers to a first approximation. And so those alleles that the lesthotroph carries that reduce the bristles are going to put those guys in danger. And reciprocally, when a planktotroph moves into a lesthotrophic population, it's bringing these costly bristles into a big egg environment. And so basically, the maternal environment outweighs the ecological environment. And whatever maternal environment is common in a mudflat creates selection for the matching zygotic alleles. And so you get this positive frequency dependence, where even if lesthotrophy would be favored by the environment, if planktotrophy is common, then a rare lesthotroph can't invade. Its alleles are mismatched to their maternal environment. And so there's this very high frequency threshold where you have to have a huge number of alleles of the opposite type moving into the maternal background for them to spread, even when they're advantageous. And so we think this is why each mudflat tends to have just one thing or the other. It's because basically that commonality of the maternal environment creates this positive frequency dependent feedback that perpetuates that thing. And so we suspect that the kind of global patterning along the coast is driven by more rare events, major storms, for example, which might facilitate mass migration or might wipe out a population and allow for invasion of a new set of colonists, basically metapopulation dynamics at the mudflat level. So that's the story I wanted to tell you, that if we're thinking about ontogeny as this process, then we need to include attention to maternal effects. And that means we have to pay attention to maternal zygotic interactions, and that can change the ecological dynamics in a way that changes the evolutionary outcomes. And as we dig into the genetic architecture in more detail, we hope to find the actual causal alleles that are underlying this process, and figure out how this process evolved in the first place and how it's maintained now in nature. And finally, this is my last slide. And so somehow I really did fail to include the slide that acknowledges Christina Zakis, former postdoc and now professor at North Carolina State, who did the quantitative genetics, and Connor Gilligan, who did the population genomics as well. So thanks to Connor, and thanks to Christina, and thanks to you all for your time.